Welcome to the mini lesson on excitation contraction coupling. As the name implies, what we will be talking about is the relationship between a stimulus arriving from the nervous system and the process of skeletal muscle contraction. What you see in the picture on the left side of the screen is of a neuron connecting to a muscle cell, and this is referred to as a neuromuscular junction. And what you see is that the neuron, the nerve cell, the very end of the nerve cell lies close to the cell membrane of the muscle cell, but there is actually a gap between the end of the nerve cell and the muscle cell. Later we will talk about the structure of the nervous system, and I will tell you about synapses. For right now, realize that the muscle must relieve stimulation from the nervous system in order to contract, and an electrical signal which is traveling along that nerve cell will jump that gap between the nerve cell and the muscle cell. Now, as you may suspect, based on the fact that the word jumps is in quotation marks, there's actually a specific process by which that happens. But for right now, just assume that the electrical signal is able to pass from the nerve cell across that gap and into the muscle cell. Now, once the electrical signal jumps into the muscle cell will actually conduct that electrical signal along its cell membrane in much the same way that the nerve cell did. This slide represents on the left the same drawing that you just saw on the previous slide, and on the right a very simplified drawing of some structures inside the muscle cell. Now, what you see on the right, labeled with the red arrows, is something called an action potential. When we talk about the nervous system, I will tell you that the action potential is a very specific type of electrical signal which is transmitted by nerve cells and some muscle cells. For right now, just think about that those red arrows as indicating the path of the electrical signal. And you can see moving from right to left in that drawing along the cell membrane, which in a muscle cell is called the sarcolemma, and you can see that labeled in the drawing. The electrical signal travels along the sarcolemma, but it also will travel deep into the muscle cell by moving down through tunnels called transverse tubules, and you can see those represented in that drawing. The thing I want you to understand is that the electrical signal does not travel in a straight line, that you can see that as the electrical signal travels along the muscle cell membrane and crosses over those transverse tubules, that the signal can spread, that the signal continues in its normal path and goes down the transverse tubules deeper down into the muscle cell. Now the next drawing is going to show you a little bit more accurate representation of the actual anatomical structures, and you can see how those transverse tubules allow the electrical signal to travel deep into the muscle cell. On the right is the picture as promised, and you can see the cell membrane, the outermost part of the drawing, and you can see the myofibrils, or the bundles of protein filaments, the actin and the myosin and some other protein filaments that are involved in muscle contraction. And what you see here, I'm highlighting with the arrow cursor, the beige line is representative of the tubule, the transverse tubule, that moves from the surface of the muscle cell deep into the muscle cell amongst the myofibrils. Now, situated right next to the transverse tubule, the slightly darker yellow or orangish color structure that surrounds the myofibrils, and you can see that it actually extends into this sort of spider web type structure here between the transverse tubules. That is a structure called sarcoplasmic reticulum. And so as the transverse tubules conduct electrical signals deep into the muscle cell, that electricity flows right past the sarcoplasmic reticulum. And that's going to be important because the sarcoplasmic reticulum, as it turns out, stores calcium ions. You might recall from when we talked about cell structure that one of the organelles inside a cell is called endoplasmic reticulum. Now, the similarity in the name endoplasmic reticulum and sarcoplasmic reticulum is not a coincidence. Sarcoplasmic reticulum is a form of endoplasmic reticulum, and if you remember that endoplasmic reticulum had a function which was to store certain chemicals inside the cell. In this case, what we're going to see is that this special type of endoplasmic reticulum, called the sarcoplasmic reticulum, stores calcium ions. And that sarcoplasmic reticulum is wrapped around the myofibrils, so that if those calcium ions leave the sarcoplasmic reticulum, they're going to immediately enter and intermingle with the protein fibers in the myofibrils. Now, you might remember when we talked about regulation of contraction that calcium plays a pretty important role in that process. So here's where those storylines begin to converge. The calcium ions, of course, coming out of the sarcoplasmic reticulum is going to play an important role in initiating that cross-bridge cycle. If you remember when we talked about endoplasmic reticulum back in the chapters on cell structure, the structure of endoplasmic reticulum, and by extension sarcoplasmic reticulum, is membranous. And it may not surprise you to see that the internal structure of that membrane is very similar to what we learned about the structure of the cell membrane, and that is that it is a phospholipid bilayer. You can see that represented in the drawing on the left. And embedded in that phospholipid bilayer are various proteins. And those proteins can serve a variety of functions. We have learned about proteins that essentially act as gates. The gate is closed. It will prevent the flow of an ion through the gate. That's what's represented in the picture right now. But that those ion channels, functioning like gates, can be opened, which would allow ions to pass through them. Remember that ions normally would not pass through the membrane because the phospholipid bilayer is impermeable to them. We also learned that these gates, these ion channels that function as gates, can be opened or closed in three different ways. The three ways that we learned that ion channels can be opened or closed is that they can be voltage sensitive, they can be chemosensitive, and they can be mechanosensitive. In this case, the ion channels in the sarcoplasmic reticulum are voltage sensitive. So they are closed until an electrical signal passes nearby, which will cause them to open. And so what we're going to see here is that electrical signal, which remember is traveling through the transverse tubule that runs in very close proximity to the sarcoplasmic reticulum, is actually going to pass right by those ion channels, and when it does, the change in voltage is going to cause the channel to open. Now you can see in the diagram that the channel is open, and the path for that little green dot, which in this case would be representative of a calcium ion, that path is now cleared, and the calcium ion can flow from the inside of the sarcoplasmic reticulum to the outside. Now remember I said that the sarcoplasmic reticulum is storing calcium, so the concentration of calcium inside the sarcoplasmic reticulum when the muscle is relaxed is very high. When that ion channel opens, the high concentration of calcium inside and the low concentration of calcium outside is going to allow the calcium ions to diffuse through that ion channel out of the sarcoplasmic reticulum and in and amongst the protein filaments of the myofibril. The term that we sometimes use when we talk about a situation where you have a high concentration in one area and a low concentration in another area is concentration gradient, and in this case diffusion would occur down a concentration gradient from an area of high concentration to an area of low concentration. At this point, we can remember what happens to calcium when it actually gets released from the sarcoplasmic reticulum and is free to move about amongst the protein filaments of the myofibrils. The diagram on the right is one that you may recall from many lessons on the cross-bridge cycle or regulation of contraction. And you can see, I will highlight with the cursor, the calcium here will bind to troponin, and when it does so, the troponin will change shape. That will cause tropomyosin to change shape. And notice the difference between the drawing at the very top, where there's no calcium present, and that tropomyosin is lying right on top of the myosin binding sites on the actin filament. When the calcium binds to troponin and causes it to change shape, and subsequently causes the tropomyosin to change shape, that tropomyosin has now slid off of the myosin binding sites, and you can see those myosin binding sites are exposed and the myosin cross bridges are free to bind, which would be the beginning of the cross bridge cycle. Once the myosin binds to the actin, the cross bridge cycle can proceed and the muscle will contract. That is when calcium is present. Now what we're going to see later is that in order to relax the muscle, the other arrow linking the top drawing to the bottom cycle shows calcium being removed, and that's the way a muscle would relax. And in fact, muscle relaxation is the topic of another mini lesson where I will describe exactly how that calcium is removed from amongst the myofibrils, and as a preview, I will tell you that ultimately we have to move that calcium back into the sarcoplasmic reticulum.